Hello students, welcome to EPG Pathshala. I am Dr. Mini Singh from the Department of Biotechnology of Punjabi University, Patiala. In this module, we will study about functional foods and nanotechnological interventions part 2, wherein we will specify about nanoformulations for value addition in foods. This module is a continuation of the previous module in which we learned about the various nutraceuticals. Not only did we learn about the nutraceuticals and their health benefits, but we also understood the various limitations which are posed in the use of these nutraceuticals and how those limitations prevent us from utilizing the efficacy or the benefits of these nutraceuticals. Can nanotechnology overcome these nutraceuticals? Is the answer that we will give in this module. When we talk about food applications, the nanoformulations are certain specific nanoformulations which can be used only in the food sector. What are these kind of nanoformulations? How they are prepared, how they are used for value addition is what we will discuss. When I talk about value addition simply means taking a nutraceutical, encasing it in a nanoformulation, reducing the size to nanoscale, making it absorbable, and adding value to that nutraceutical. That is the reason why we call value addition. Furthermore, value addition has another connotation. One, let's say we have prepared a nanoformulation, but do we consume it as it is? If we consume it as it is, that is a nanoformulation, but we can also use it in the form of mixing it in other form of foods. During the course of this module, we will see an example where we have used the Betacarotene nanoformulation and used it for enhancing the value of butter. That means we have now a butter which has got a provitamin A capacity in it. This module, therefore, has been designed with the following objectives. The various approaches to overcome the limitations in the bioavailability of nutraceuticals. We will also discuss about the types of nanoformulations which are most accepted for food nanoformulations. Not all kinds of formulations can be used in food applications merely because of their biocompatibility. So it is important that we choose only formulations which are biocompatible, which means they are in sync with the biological system and the biological system does not reject those formulations. We will take two specific examples in this module. One will be the formation of beta-carotene nanoemulsion and how that has been used for value addition of butter and juice. And the second example will be specific, in which we have extracted certain bioactives from peels of a fruit and converted those bioactives into nanoformulations and have converted it into a nutraceutical product. We will also wind up this module by making certain considerations before commercialization. It sounds simple that we have prepared a nanoformulation and now it is ready for commercial purposes. However, there are certain considerations towards toxicity testings, considerations towards certain FDA regulations. So before we can think about commercializing any of these formulations, it is important to keep those considerations in mind. From the previous module, we understood the limitations which are posed by most of the nutraceutics. Let us go over them once again. 1. They are poorly aqueous soluble, which is attributed sometimes to their structure. 2. They have low membrane permeability because they do not have receptors on the intestinal cells. 3. Instability in the GI tract, which means that they get conjugated or they undergo reduction. Bringing your focus again to the three channels, channel A, channel B, and channel C. These nutraceutics, when consumed, will either just pass through the gut into the feces, which is channel A, or part of them may be absorbed by the intestinal cells, but they will undergo premature metabolism, or they will enter the liver and yet undergo a phase 2 premature metabolism. Very few, which are very actively bioavailable, will enter into circulation. So what do we need to do to make them bioavailable? Various approaches have been reported to overcome these limitations. The first of being the use of adjuvants. The most popular adjuvant which is used is piperine. What does piperine do? It inhibits the glucuronidation and formation of non-bioactive conjugates That means the conjugation process that we had studied in the previous module, that is glucuronation, sulfonation, it inhibits that process. The reports have also shown that there is a 20 times increase in the bioavailability of certain nutraceuticals when used in combination with piperine. However, piperine may lead to the biotransformation of these bioactives into derivatives, which means they may lower the functionality of these bioactives. Another method to overcome the limitation is by inhibition of P-glycoproteins. P-glycoproteins are present in the intestinal cells and they are efflux proteins, which means that by passive diffusion, if some of these molecules by way of passive diffusion find their way into the cells, the P-glycoproteins will efflux them back outside into the lumen. Therefore, if we are able to inhibit the P-glycoproteins, it will only mean that we are enhancing the absorption of these bioactives. One compound, which is called verapamil, it is a PGP blocker. It inhibits the P-glycoprotein and blocks the efflux of curcumin. This is well reported in literature. However, since verapamil is a drug, therefore it cannot be used in preparation of functional foods, which means that these inhibitors, PGP inhibitors, are mostly drug molecules and they cannot be used for developing functional foods. Yet another way of overcoming the limitations of low bioavailability is by the use of phospholipid complexes. Phospholipid complexes are efficient delivery systems for lipophilic compounds. They provide stability to the molecule in the GI tract. This by far has been the most common way of enhancing the bioavailability of hydrophobic compounds. Despite the presence of these three modes of enhancing the bioavailability, they do have their own specific limitations, such as non-use in food materials. So, in the light of this, can nanoformulations be used for overcoming these limitations? The answer is yes. They enhance the colloidal solubility of bioactives. The presence of specific clathrin and caveol receptors leads to internalization of nanoformulations. The protective layer prevents degradation and biotransformation into non-bioactive metabolites. From where does this protective biolayer come? When we prepare nanoformulations, we are using lipids or polysaccharides as carriers. It is these lipids, polysaccharides, and surfactants which render a protective layer outside the bioactive. Therefore, not only does it provide the nanoformulation a protective layer, but it also leads to specific clathrin and caveol receptors, which means that nanoformulations show a double-pronged action. We understood this diagram in the previous module, yet once again, now since we are more clear about the nanoformulations, role of nanoformulations, let's go through it once again. We have been talking about clathrin and caveol mediated endocytosis, and now the green dots that you see are the encapsulated bioactives, encapsulated either by lipid carriers or by polysaccharide carriers or even by protein carriers. This will be understood in our next module. When the bioactives are encapsulated within these nanoformulations, they receive the acceptability by the intestinal cells merely by active diffusion or by caveol and clathrin mediated endocytosis. How does that happen? When we say nanoformulations, we are talking about reducing the size within a certain range. The FDA has classified that range from 1 to 1000 nanometers for food particles. The image shown here shows the size-dependent absorption characteristics of particles. The larger the size, the poorer the absorption. As we reduce the size to nano dimensions, there is maximum absorption. This is because of enhanced surface area per unit mass of the nanoparticles. So they are expected to be more biologically active than larger size particles and show maximum absorption rate. We have now understood what happens when we make a nanoformulation. We encase a bioactive with something and then we reduce the size. What happens with this is the absorption in the body will increase. So what are these types of nanoformulations which are being used or which are being prepared for value addition of foods? The three most common kind of the most acceptable kind of formulations are number 1, the nanoemulsions, number 2, the liposomes, and number 3, the solid lipid nanoparticles. There is a specific reason for the high acceptability of these three particular kinds of formulations, and that reason is it uses natural lipids mostly which are edible for use. So when we use a lipid molecule, whether it's a liquid lipid, if we use a liquid lipid, we are essentially making a nanoemulsion. If we use a solid lipid, we are talking about a solid lipid nanoparticle. So a lipid carrier which is in sync or which has got high biocompatibility with the human system, all these three lipid carriers are highly used or accepted in the making of nanoformulations for food applications. The two lesser known formulations are number 1, polymeric, and number 2, nanodispersions. Polymeric nanoparticles essentially use certain polysaccharides such as PLGA or PLA. PLGA and PLA have been approved by the FDA for use in foods. However, they do suffer from the limitation of low loading of the nutraceutical. Scientists are working on this, and once approved, this will be a very effective carrier in the near future. Nanodispersions are lesser used in food nano applications for the reason that nanodispersions during their preparation use solvents, organic solvents in the process. Although these solvents are removed subsequently or they are rotary evaporated, but there is every possibility of certain traces being left behind. So in terms of food safety applications, we do not want any sort of residue of an organic solvent in the food. Therefore, these are slightly lesser in prominence. Of the different kinds of nanoformulations which are popularly used for use in foods, nanoemulsions are the most popular kind of formulations. In nanoemulsions, the carrier is essentially lipid based. So we have the oil with the bioactive, there is a surfactant, and there is water. The surfactant essentially reduces the interfacial tension between the oil and the aqueous phase. There are two ways of preparing nanoformulations: the high energy methods and the low energy methods. The high energy methods are essentially the high pressure homogenization and ultrasonication. The figure depicted here, the upper half of the figure depicted here shows the preparation of an oil and water nanoemulsion, the starting material being an oil and water macroemulsion in the presence of a surfactant. The two high energy methods are ultrasonication. So when this macroemulsion is passed through a sonoprobe renders high intensity sound waves which reduces the size of these nanodroplets, making them nano in size. Whereas, on the other hand, the high pressure homogenizer has a channel through which the macroemulsion passes, and due to high pressure, the size breaks down into nanoformulations. The low energy methods are the emulsion inversion points and the phase inversion temperature. In the example depicted in the figure, example B, is a water in oil macroemulsion which undergoes a phase inversion at a certain temperature, wherein water in oil becomes an oil in water nanoemulsion. Similarly, in an emulsion inversion point, the water is added into the emulsion to a point where water becomes the continuous phase and oil remains as the dispersed phase. So this phase inversion leads to formation of nanoparticles. Looking closely at how a nanoemulsion droplet looks like, it consists of a lipid phase containing the bioactive, which is dispersed in a continuous aqueous phase. With each oil droplet being surrounded by a thin interfacial layer consisting of an emulsifier molecule with droplet size in the nanometer range. The figure depicts the central core, which is hydrophobic, and the outer polar head of the surfactant and the non-polar tail of the surfactant, all of this dispersed in an aqueous phase. Among the various nanoencapsulation approaches, nanoemulsions are generally suitable and widely accepted for food applications. The reason for this is the use of carrier molecules which are edible. Mostly edible oils are used for preparation of nanoemulsions. What is shown here is a TEM image of a nanodroplet in an emulsion. Very clearly, we can see a central dark core, which is the lipid core, surrounded by a gray layer, which is the layer of the surfactant, and these nanodroplets are dispersed in a continuous phase, which is water. The figure on the right is a magnification of one of the nanodroplets, in which the dark spots show the encapsulation of the bioactive. Since we can see there are no black spots in the outer aqueous phase, it is clear that the bioactive is confined or encapsulated within this capsule. Apart from nanoemulsions, liposomes, which are spherical vesicles arising from self-assembly of a phospholipid bilayer membrane, are another key components of nanoformulations. This formation of liposomes is merely based on self-assembly. Shown here is a structure of a liposome containing a hydrophobic drug or a bioactive, which we've been talking about, and a hydrophilic drug which is in the core. The aqueous compartment engulfs the hydrophilic drug, whereas the bilayer engulfs the lipid bilayer. These phospholipids, therefore, as is clear, have tubular shapes owing to the presence of two acyl chains attached to a polar head, and the hydration results in a bilayered membrane. The bioactive is usually intercalated within the lipid bilayer. This sort of formulation has a double strong action. It can not only encapsulate the hydrophobic molecules within the membranes, but also enhance the efficacy of hydrophilic molecules. We've just studied about liposomes and how they are used as these bilayered molecules are used as vehicles for adding value to foods. Their specific advantages are increased stability. They are bilayered, therefore their stability is much enhanced. Furthermore, they're biocompatible, they're completely biodegradable and non-toxic. Another popular kind of formulation in the nano dimension are the solid lipid nanoparticles. The difference in solid lipid nanoparticles from other carriers is that the core is made up of a solid core, a solid lipid. So the colloidal carrier system is composed of a high melting point lipid as a solid core coated by an aqueous surfactant. Since it is a high melting point liquid and does not melt under ambient temperatures, therefore it maintains the hard structure of the core. In solid lipid nanoparticles, therefore, the lipid which was earlier liquid in a nanoemulsion is replaced by a solid. These lipids, solid lipids, include triglycerides, partial glycerides, fatty acids, hard fats, and waxes. A significant advantage of these solid lipid nanoparticles is their stability, which is owing to their solid character. The image we see now is a TEM image of a solid lipid nanoparticle. You can see very clearly this is a classical textbook image of a solid lipid nanoparticle in which the core is very dark, which is exemplifying a solid core of the particle. This solid core is surrounded by a whitish layer, a slightly gray-white layer, and that layer is that of a surfactant, and the outside is the continuous phase. So this is an oil in water solid lipid nanoparticle dispersed in an aqueous medium. A specific advantage of solid lipid nanoparticles is their stability, and this heightened stability is attributed to the solid lipid. The liquid lipids will tend to melt under ambient conditions, especially under certain temperate climates. However, a solid lipid does not undergo these changes and is able to maintain its physical state. Secondly, it has a very high payload, which means the entrapment efficiency or the encapsulation efficiency may range up to 95 to 96%, which means that we'll be able to achieve that bioefficacy at a much lower dose. Other than that, it is also biocompatible in nature, which lends it a further advantage. The figure shown in this slide summarizes the use of lipid nanocarriers, the liposomes, nano emulsions, and lipid nanoparticles, which are emulsions, which is clear again that the liposome contains a bilayer vis-a-vis a nanoemulsion and a lipid nanoparticle, which are monolayered. The liquid lipid has a liquid center, whereas a solid lipid has a solid core. The slightly lesser important formulations in food applications are nanodispersions. The reason for their low popularity is because of the use of solvents in their preparation, and solvents are not preferred sources for use in foods. However, let's see how these nanodispersions are made. Solvents are used to dissolute the hydrophobic bioactives. In this example, we can see the use of acetone plus a polymer plus a beta-carotene, which is the bioactive which is being encapsulated. This is mixed drop by drop into an aqueous phase which contains water and an emulsifier. And much like what we understood in the lipids, here we have the bioactive within a solvent. After this mixture is prepared, the solvent is evaporated and the nanoparticles are generated. However, since there could be the possibility of presence of even traces of solvents, therefore these kind of formulations are not favored in food applications. Seen in this figure is a TEM image of a droplet of a nanodispersion. As told before, nanodispersions involve the use of organic solvents and therefore slightly lesser used. But the purpose of showing this image to you is for you to understand how the nanodispersions are dispersed in the aqueous phase. All of these formulations, whether it is nanoemulsions or solid lipid nanoparticles or nanodispersions, will appear in the form of these nanodroplets in a continuous phase. We will now see two specific examples of value addition of food through nanoformulations. The first being a beta-carotene nanoemulsion prepared by high energy emulsification evaporation technique. In this process, beta-carotene, the top figure on the left, you will see particles of beta-carotene in water which are not soluble, whereas on the right is a nanoformulation of beta-carotene which is completely soluble. The figure on the left below shows three plates. On the right is again beta-carotene particles, flakes dispersed in water, which is not dispersible. On its left, you will see beta-carotene dispersed in oil, which is only partially soluble, and as a nanoformulation on the picture shown on the right. This work involved the preparation of a nanoemulsion using olive oil as a carrier with a surfactant molecule and subjecting the macroemulsion to ultrasonication for a period of 30 minutes. After 30 minutes, there was reduction in the size of the molecules of the nanoformulations to nano dimensions. In this case, 122.6 nanometers. When this nanoemulsion was dissoluted in water, as you can see on the figure on the right, it was completely dispersible in the aqueous system. The example shown here was a work done by my research team. We extracted bioactives from wastes. These wastes were collected from an industry which is processing mandarins into juices. This is a government-based industry, and this is a collaborative project which is being run by SELF and the industry partners. The objective of this project was to utilize the bioactives which are present in the peels, and the reason why we thought that these bioactives would be present in these peels was merely because of the color of the peels. We have been understanding right from the beginning that brightly colored foods obviously mean that they will have abundance of bioactives. In this case, we thought that these bioactives present in these peels would be the classes of limonoids, carotenoids, and flavonoids. In the course of the work, we were able to identify different types of limonoids, whereas as well as carotenoids, as well as flavonoids present in these peels. Thereafter, we set out to overcome the limitation of low aqueous solubility, because all these classes of compounds we have also studied before are hydrophobic in nature, which means that even if we try to use it, we will not be able to utilize the efficacy of these bioactives. Therefore, we prepared nanoformulations. What is shown here in the plate is a dried powder of these peels, which was dried and stored under refrigerated conditions. It is these powders which are used for work in the lab. Next to it is a vial showing the extract from these powders. This extract of the peel powders contains the bioactives which are limonoids, carotenoids, and terpenoids. However, this extract cannot be used or is not efficacious enough because it has all the hydrophobic components. In the next step, we prepared solid lipid nanoparticles of this extract, and as you can see in the picture, the solid lipids containing the bioactives in it, and they were finally packaged into a capsule which makes a nutraceutical. Not only was it packaged into a capsule, but it was also made into a liquid nanoformulation, which is now called Limopan. Limopan is presently undergoing human clinical trials. What we are going to demonstrate to you is how nanoformulations help in value adding. That means we've studied in the modules that there are limitations of aqueous solubility which further lend to limitations in bioavailability, and how nanoformulations, different kinds, whether they are nano emulsions, solid lipid nanoparticles, or nanodispersions, how they enhance the aqueous solubility. On the extreme left is an extract. It is a nutraceutical extract. This was extracted from peels of mandarins, and this extract contains hydrophobic compounds like limonoids, carotenoids, and flavonoids. And what we will see is that this extract, let us see for ourselves whether this extract dissolves in water or not. We will take a small volume of the extract and disperse it in an aqueous system. What we see is the non-aqueous solubility of this system, wherein the extract does not go into the system and only remains on the surface. We prepared 3 kinds of nanoformulations with these extracts. The first is a nano emulsion. The second is a solid lipid nanoparticle. As you can see, there is a difference in the physical structure. The nano emulsions are liquid because they are comprised of liquid oils, as against solid lipids, which have a solid lipid as their component. The third kind of nanoformulation is a nanodispersion, which we had also studied are lesser used forms of formulations, merely because of the use of solvents in the same. So let's see how these nano emulsions, whether these nano emulsions are able to dissolute in an aqueous media. As again, we will take a small volume of the nano emulsion and see whether this disperses in the aqueous media. What we see is complete dissolution of the extract in the form of a nanoformulation against an extract which is in a native state. This is how we can enhance the bioavailability by enhancing the aqueous solubility of hydrophobic compounds, hence lending character to nutraceutic nanoformulations. We have clearly seen how the use of nanoformulations helps us to overcome the limitations of bioavailability, aqueous solubility, and how we can use nanoformulations in overcoming the limitations and using these in preparing functional foods. However, it is important for us as scientists to know that we cannot just simply use them without certain checklists. The first, the FSSAI regulations, the Food Safety and Standards Authority of India. The FSSAI draws its regulations from FDA. The FDA, as told before, classifies food nanoparticles into two categories, one within the 1 to 100 nanometer range and the other 101 to 1000 nanometers of range. The reason why FDA is thinking about the safety assessments or the toxicity testing of these nanoformulations is because there is always a certain fear that if the nanoparticle goes below 100 nanometers, it may cross the blood-brain barrier, and we are not in a position yet to tell what kind of toxicity it may impart to the brain. Therefore, to follow the FSSAI regulations, the FDA regulations, and to perform proper toxicity testing are the prerequisites before we can think of applying any of these nanoformulations into functional foods. So we learnt in this module how value addition can be done through processing. When we say processing, we are talking about making nanoformulations. Nanoformulations could be nano emulsions, nanodispersions, solid lipid nanoparticles, or polymeric nanoparticles. By far, the most biocompatible nanoformulations are those which have got lipid nanocarriers in them, and this is what lends success to the use of nano emulsions, solid lipid nanoparticles, and liposomes. Lipid carriers are biocompatible, and that is the reason why of their high use in food applications. These value-added products will add a great value to the food market. However, we have to keep in consideration the FDA regulations, the toxicological studies, and these value-added products will in the near future add a whole new revolution in the arena of nutraceutic nanotechnology. Thank you.